Okay, so I'll be talking about a lipoarabinamano assay as a pharmacodynamic biomarker. I'll be presenting some of the data that we presented in a recent critical path innovation meeting, but the primary focus of my presentation will be on that CPIM and how it went for us and how I would hope it would go for just about everybody who's been presenting on potential new tools. So what is a critical path innovation meeting or a CPIM? Some of what I'm presenting here, Anne's going to go into a lot more detail on it later, and so I won't be talking too much now. But they're administered through the FDA's Office of Translational Science, which is within CDER, the Center for Drug Evaluation and Research. The CPIM is intended to be very broad in scope and serves as an opportunity for general discussion. It's to foster discussion of the science and the regulatory aspects as well in drug development. And anyone who has a potential drug development tool can request a CPIM, and my understanding is that actually the Office of Translational Science is getting an awful lot of requests for them these days. It's an assembly of appropriate FDA experts from CDER offices and other centers. In our case, we also had representatives from CDRH. The meeting discussions are non-binding, and again, it's to be a rather informal exchange. The potential biomarkers can be discussed, emerging technologies, novel clinical trial designs, and one of the things very clear is that the CPIM is not meant to provide advice or a discussion of a regulatory pathway for a particular assay or diagnostic. Just getting into a lot more detail, which I think Anne is actually going to get into. So here is the timeline that we experienced. Was in November we put in the CPIM request. Mid-December we heard back that it had been received and that the request had been accepted. Then in January our date was confirmed, and we had the meeting in March. We expect to hear back sometime in May, and the input that we got from this meeting will be used to inform us in the development of future plans. Before I get into what we presented at the CPIM, one of the things I want to really emphasize is that, as it said in the earlier slide, that this was meant to be an informal meeting, an exchange of ideas, a scientific discussion, and coming from a pharmaceutical company and having interactions with FDA and EMA, I'm like, yeah, it's going to be relaxed and it's going to be informal and it's going to be somewhere on the level of and looking at regulatory colleagues. I won't say it's on the level of a root canal, but I didn't expect it to be what it was, which it was informal. It was an exchange. We got a lot back. I learned a lot about the questions that were asked, that A, we weren't communicating, or B, we didn't have the answers yet, and these were important things that we needed to get into. And I hope the representatives from the FDA felt the same way about the exchange that went back and forth. And it really was an exchange, and I think it was quite productive for us and actually almost even enjoyable. So what I'm going to be presenting next were the slides that we presented during the CPIM. I've taken a few of the slides out about the regulatory approaches. So we presented in the meeting that the opportunity was that there's a high unmet medical need for a real-time assessment of efficacy in TB drug development trials, and talked about the different kinds of trials, and kept pointing back to that the outcome measures in most of these early trials were microbiologic based, that there's a delay in getting the microbiologic information, and that added to both the time and the complexity of carrying out the trials. We talked about the early bactericidal activity trials, the design itself, how it was conducted, using qualitative colony counting or time to detection as measures of the bactericidal effect. We contrasted the difference between quantitative cultures and time to detection and some of the differences that we saw there. Talked about sputum culture conversion, its status within the guidelines, and that it's mentioned now in the US guidelines as well as the European guidelines as a potential endpoint to justify an accelerated approval or conditional approval in Europe. We discussed the EBA as being difficult to conduct, that it requires the colony counting, which takes 4 to 6 weeks to get back colony counts, that the quantitative culture is requiring serial dilutions, that it's a highly technical and complex process. It's not easy to conduct. And also with midget TTD requires a separate processing stream so that if you're going to use both, that you have essentially 2 streams to process your specimens. We mentioned that it is complex, and in fact there are a limited number of laboratories in the world that can conduct EBA studies, and most of those that have been submitted to regulatory agencies have been done at a single site in South Africa. And so that there's the potential for a bottleneck there. Also relying on either solid or liquid media cultures, you have the problem with losing data due to contamination, and drug carryover can also, is at least, at the very least, a theoretical problem, and it is mentioned in the European guidelines. Again, with time to detection, it has its own challenges and its own limitation, not the least of which is that there is good evidence, and actually we're generating some more evidence, that time to detection changes over time. That time to detection is a function of both the bacterial load and the metabolic state of the bacilli. And if you're changing the metabolic state of the bacilli with the drugs that you're giving, there is a time factor that you have to build into time to detection for the bacilli to normalize its metabolism and then begin replicating, consuming oxygen, and giving you a positive output. Because of that, and until we understand better the nature of the relationship between the number of bacilli and the metabolic state, and what the metabolic state is after a certain period of treatment with different drugs, really what TDD is going to give you is really more of a qualitative assessment of whether or not things are declining, and a qualitative assessment of the rate, but it's not going to be a quantitative assessment in the same way that a quantitative culture would be. Again, went through sputum culture conversion and talking about the challenges there, particularly, again, with contamination and the potential for drug carryover. Rather complex chart here, but laid out a traditional approach to doing regimen development, so that you start out with 5 regimens and you do a regimen EBA study. You then wait for the culture results to come in, you analyze that data. You then design a phase 2, 2-month sputum culture conversion study. Let's say 3 of your regimens make it through to that phase 2, 2-month SCC study. But I also pointed out that at least in our experience, our greatest time sink in the clinical development pathway is often the regulatory time between trials. And that in some of the countries where we have worked, where we might be less likely to work again, the time delay has been up to 12 to 18 months. Some of that can be political factors, some of that can be just the capacity of the regulatory authority to deal with a clinical trial application, and that adds a great deal of time to the total clinical development pathway. So the traditional approach coming out of the 2-month sputum culture conversion study would be the selection of a regimen to go into a phase 3 study to be compared with your active comparator. The suggestion that we presented to them, or the opportunity that we presented to them, would be the idea of doing either seamless phase 2, phase 3 studies, or really building into a phase 2C and going into phase 3 studies, where you might start 5 regimens at once, that you would be using the real-time assay that would give you same day you get the sputum, you also get a quantitative assessment of the bacterial load in that sputum, that you would have to have well-defined stopping rules. Those stopping rules could be based upon going below the proportion of patients who are going with producing specimens that are below the lower limit of detection. A stopping rule could be based upon the rate of decline that you need to see a 2-log decline or a 3-log decline over a 1-week or a 2-week period in order to define that regimen as being successful and keep it in the study. In this case, 4 of the regimens hit that stopping rule and are eliminated, and then the 3rd regimen would be carried on for evaluation of a pivotal endpoint. Not listed there is the active comparator, which might be HRZE as kind of a standard comparator these days. And one of the other points that we made is that evaluation of the pivotal endpoint is that we were not suggesting that this LAM assay right now should be a pivotal endpoint. And I think this was an important point that people within, from the FDA within the CPIM, that when we were clear about that, when we got that across, that we would still be assessing the efficacy of the regimen in comparison with the comparator regimen on currently accepted measures of what an efficacious regimen is in a final endpoint, which in trials that we've done has been proportion success, which is the portion achieving sputum culture conversion by a certain time point, maintaining that conversion over a duration of therapy, and then remaining culture negative for a certain period of time after the end of treatment. We wouldn't change that. That would still be the final evaluator. That what we would be doing is allowing for the evaluation of a number of regimens all at once. Because we have the real-time assessment of quantitative load, we'd be able to either to drop them out and end up with just 1 or hopefully even 2 regimens that would make it to the end. We also pointed out that this is something that was still going to take a lot of work. There are a lot of biostatisticians in the world who will make a living off of advising how to do this. And this itself, as it was noted, a CPIM can be, and a qualification can be arranged around the design of a clinical trials paradigm. And so this might then lead into another work stream. Do you hear that, Deborah? Another work stream coming up. And a separate qualification process. So a little bit, then we got into, after talking about the opportunity, we got into talking about the assay itself, that we have developed monoclonal antibodies that detect liporabinomannane, which is a major cell wall component of Mycobacterium tuberculosis. It's highly specific for LAM from Mycobacterium TB and a few of the slower growing mycobacterial strains, but we haven't seen a lot of problem with the sensitivity or the specificity of the assay in the specimens that we've dealt with so far. And a lot of this we showed data that was in the briefing package, and I don't have here in the slides, but we did show data that shows that there's no cross-reactivity with the oral bacteria, which has been a limitation of LAM assays working in sputum. We also presented data that there's a strong correlation between sputum, LAM, and the CFU counts and the time to detection from other assays. We showed data that we think supports the idea that the LAM assay is not affected by contamination. We have results giving quantitative loads, quantitative estimates that correlate with a time to detection when the agar media culture was contaminated, and vice versa, that correlated with the agar media culture when the liquid media culture was contaminated. So we believe that it's not affected by contamination. It's a LAM ELISA. It takes about 20 minutes for the LAM extraction and about 5 hours for the ELISA itself, using the current platform. And that platform we're trying to optimize and have a target to eventually have it to be less than an hour. The assay detects about 1 picogram of LAM for every approximately 8 CFUs. This was established in an in vitro assay using H37RV. We need to see if this is the same across different strains. Our lower limit of detection is about 8.5 picograms per ml, which would be about 69 CFUs per ml. Lower limit of quantification, 15 picograms, or about 121 CFU per ml, which is in the range of an agar media culture. It's not as sensitive as midget, but we think is as good as, or maybe a little bit better than, agar media cultures. As I mentioned, specific for lamb from mycobacteria with lower sensitivities to the slow-growing NTMs. So we summarized 3 studies, one that was in smear and culture positive sputum specimens, where the sensitivity was 100% in 2 of the studies. One of them had 70 specimens collected, and the other 100 that came from a biobank. In a study where there were smear negative and midget positive sputum specimens, the sensitivity was 57% in the smaller group, which was of 57, and in one study, and then 74% from the biobank studies. An issue there is that smear negative and midget positive, and what the bacterial load is in that midget positive specimen. The specificity was 100% for those that were judged to be not have tuberculosis, and 28 of the cultures grew out non-tuberculous mycobacteria, and we got a positive lamb signal in 2 of them, or about 7%. We also did a 4th study where we looked at changes in lamb over 56 days. We did smear on the raw sputum. We did both the LAMP, which is a nucleic acid amplification test, and lamb ELISA on homogenized sputum, and then we did time to detection in midget using sediments from those specimens. And so we show here in the left column that the lamb showed about a 1.24 log decline over a 14-day period. We contrasted that with a 1.58 log decline in the EBA study where we were evaluating delaminid, but these results are for the HRZE regimen. And in both cases, patients were being treated with HRZE. So similar magnitudes of decline over a 14-day period, and then the qualitative result of 227.8 hours change over the 14 days. So everything changing in the same direction. We compared, this is, and I get messed up on this a lot, and a lot of other people get messed up on it when they look at it. On the y-axis, we have the percent positive cases. This is not the amount of lamb we're detecting or the number of bacilli that we're detecting. It's the percentage positive cases. And so you see very quickly in blue that the proportion positive on the AFB smear declines very quickly and then changes slowly over time, but there's not a wide range of detection there that you could use to monitor changes over time. At least with the LAMP, with that nucleic acid amplification test, there's some decline early on, but it remains positive throughout the 56-day period, most likely because it's amplifying DNA from dead bacilli, and you're not seeing the quantitative change. For both the lamb ELISA test and for the midget test, there is some dissociation at 14 days and 28 days. The lamb ELISA seems to be declining more quickly in terms of the proportion positive, but again, by the time you get out to 56 days, there's a very, very good matchup in the proportion positive. We have data that's coming from another study that we were able to share some preliminary results, but as preliminary results from somebody else's study, we aren't able to share them here. But this is called the NextGen study, sponsored by the NIH, being conducted in South Africa. They're looking at 8 different regimens, or 8 different drugs or regimens in an EBA design. So they're looking at HRZ, moxifloxacin as individual drugs, then HZ, RZ, MRZE, and HRZE. They're doing quantitative cultures. They're getting midget time to detection. They're evaluating a number of other potential immunologic biomarkers, and they're also looking at changes in imaging, PET CT scans, and they've nested the lamb ELISA within this, and we'll be getting the data back from them on this so that we'll be able to compare and contrast the lamb ELISA with all of the other potential biomarkers that they're evaluating. So next steps is that we need to obtain the final data from the next-gen study. We're in the process of studies of degradation of lamb during treatment to perhaps answer the question as to why do we believe we're only detecting viable bacilli? The question comes, well, don't the drugs change the conformation of lamb? But not kill the bacilli, but still, since we're correlating so closely with the quantitative counts on culture, so that's something that we need to explain. And we're also in the process of trying to identify lamb epitopes for where the antibody is binding, which again may help explain some of why we believe we're only detecting the viable, or maybe it's just culturable bacilli. The context of use has been mentioned, and that's really the heart of the qualification process, or the launching point for the CPIM. And so we've went around with the context of use, and so this is ours, of lamb being a pharmacodynamic biomarker for quantitative measurement of bacterial load in sputum, that the pharmacodynamic biomarker should be considered with other microbiologic measurements, such as culture, to evaluate a real-time treatment response and give some examples of where that might be valuable. This is a living document until you actually get qualification. I'm sure we will change this over time. Summary, we believe that we have an assay that's valuable, that it measures culture, and that it measures bacterial load in the same way that culture does, and it's not affected by contamination. I was supposed to have a slide that showed the entire study team. It's very large, brought together by CPTR, and I think we were interested in this, but I don't think we would have been able to get off the dime and evaluate this lamb assay in the way that we did without the assistance and support that we got out of CPTR and in general, and from the study team in particular. Lots of good questions and valuable advice. So that's that.